Okay, Professor Tabs is back. So we are talking about antigen presentation, and we have discussed the basics of how a dendritic cell would present antigen to a T cell, and how we should recognize the class 1 MHC are corresponding to CD8 and CD8 cells, and class 2 MHC corresponding to the Cd4 cells. And we have talked about the differences between dendritic cells, B cells, and the macrophages basically. So let's continue on. Remember that blood-borne antigens are captured by antigen-presenting cells in the spleen, whereas the lymph node captures antigen from epithelium and connective tissue. Very important to recognize that. And we know that the key presenter of antigens to naive T cells is the dendritic cell. And we quickly reviewed in the last video the antigen uptake, antigen presentation, and the general response. And we should recognize, of course, that for dendritic cells, it results after the antigen is presented in naive T cell activation, which results in clonal expansion and differentiation into effector cells. But for the macrophages, this would result in effector T cell activation of macrophages, and this is cell-mediated immunity. And for the B cells, we have effector T cell activation, which results in B cell activation and antibody production or humoral immunity. Wow, you like it? My eyes get big like that. Anyway, continuing on. So let's talk more about the properties of antigens which are recognized by T cells. So just as a basic review, remember one of the differences between the B cells and T cells is that the T cells are specific for amino acid sequences of peptides and recognize and respond to these sequences only when the antigens are attached to APCs, whereas the B cells and secreted antibodies bind soluble antigens in body fluids as well as on the surface. So it's not just the proteins or the amino acids that are being recognized by the B cells. They have a much wider range. But the T cells are recognizing the peptides. Remember that the T cells recognize foreign peptide antigens only when these peptides are bound to and displayed by the MHC molecules. And this is known as what? This is known as self-MHC restriction. Okay, so now let's talk about the properties of antigens recognized by T cells. So how about Cd4 cells, Cd4 helper T cells? They recognize peptides which are bound to class 2 MHC molecules, and these peptides are derived mainly from extracellular proteins that are internalized into the vesicles of APCs. How about the CD8 or cytotoxic T lymphocytes? These recognize peptides which are bound to class 1 MHC molecules. And remember that these peptides are derived mainly from cytosolic, usually endogenously synthesized proteins. So the big takeaway here to remember is that the Cd4 cells are bound to class 2 MHC molecules, whereas the CD8 cells are bound to class 1 MHC molecules. So here's a question here. What are the class 1 molecules? Where are they expressed on? They are expressed on all nucleated cells, except for, of course, the red blood cells because they do not have nuclei. But the class 2 molecules are expressed mainly on dendritic cells, macrophages, and B lymphocytes. So let's think about how class 1 and class 2 associated antigen presentation influences the nature of the host T cell response. So as a summary, we can say that by segregating the class 1 and class 2 pathways of antigen processing, the immune system is able to respond to extracellular and intracellular microbes in different ways that are best able to combat these microbes. So let's quickly think about the action of the class 2 MHC. associated presentation of extracellular antigen to helper T cells. What happens with the antigen uptake or synthesis, and then the antigen presentation, and then the T cell effector function? What happens? So with a macrophage, for example, plus the extracellular antigen, we would have the result is the CD4 helper T lymphocyte releasing cytokines. Then with the macrophage activation, you'd have the destruction of the phagocytosed antigen. And with an antigen-specific B cell, we would have the extracellular antigen, and then the result would be cytokines released with the CD4 helper T lymphocyte, and B cell antibody secretion will result in the antibody binding to the antigen. How about with the class 1 MHC and the associated presentation of cytosolic antigen to cytosolic T lymphocytes? Remember, with the class 1 MHC, we are talking about the CD8 cytolytic T lymphocytes. So in this case, the class 1 MHC and the endogenously synthesized antigen would result in the CD8 cytosolic T lymphocytes killing of antigen-expressing target cell. So there we have basic explanation of class 2 MHCs and class 1 MHCs. So a quick true-false question. Specific T cell activation requires peptide antigens to be presented in the context of MHC. Is this true? Yes, this is true. Remember that the T cells are a lot more picky than the B cells. The T cells prefer the peptide antigens. Another MCQ. Cells that display MHC-associated peptides are called what? A. Red blood cells. B. Dendritic cells. C. T cells. D. Platelets. And of course the answer here is B. Dendritic cells. What are two other dendritic cells? We have B cells and of course macrophages.